My name is Christine Brown. I'm a professor and deputy director in the T-cell Therapeutics Research Laboratories at City of Hope in Duarte, California. On behalf of my co-authors, it's a pleasure to present our phase 1 study, which is being conducted to evaluate CAR T-cells incorporating a chlorotoxin tumor targeting domain for the treatment of recurrent or progressive glioblastoma that expresses matrix metalloproteinase 2 or MMP2. Glioblastoma is the most common malignant brain tumor and is known to be highly aggressive with substantial inter and intratumoral heterogeneity. The 5-year survival rate of patients diagnosed with glioblastoma is approximately only 5%, and median overall survival following recurrence is just 5 to 8 months. Currently, no standard of care therapies are established for recurrent glioblastoma, and more effective therapies are greatly needed. Glioma targeting CAR T cells have shown anti-tumor bioactivity in ongoing clinical trials, but response rates are limited due to the challenge of tumor heterogeneity. In this phase 1 study, we're investigating the safety of a novel chlorotoxin-directed CAR T cell therapy in patients with glioblastoma. And this CAR platform was developed by my laboratory at City of Hope. Chlorotoxin CAR T cell therapy uses a novel tumor recognition domain, the chlorotoxin peptide, that identifies and binds to a multi-receptor complex comprising MMP2 that is expressed by glioblastoma cells. In vitro studies, chlorotoxin CAR T cells have demonstrated strong binding potential to a broad spectrum of glioblastoma cells. Moreover, preclinical studies suggest that chlorotoxin CAR therapy is potentially safe, showing negligible killing of normal human cell lines, no adverse effects following either regional or systemic administration in mice, and no off-target recognition of normal human or murine cells. We have also shown that chlorotoxin T cells mediate potent in vivo anti-tumor activity. Following single intracranial intratumoral or ICT administration of chlorotoxin CAR T cells in orthotopic glioblastoma mouse models, potent in vivo anti-tumor activity, including tumor regression and significantly improved survival, was observed. Based on this promising preclinical data, we've initiated a first-in-human phase 1 study. This is a single center feasibility safety study to evaluate adoptive transfer of chlorotoxin CAR T cells for the treatment of patients with MMP2 positive recurrent or progressive glioblastoma. The primary endpoint of safety is assessed by evaluating dose-limiting toxicities, cytokine release syndrome, and all other toxicities. We will also determine the maximum tolerated dose and recommended phase 2 dosing plan. This study will enroll patients with relapsed grade 4 glioblastoma that are 18 years old and above, with a Karnofsky performance status of at least 60% and MMP2 expression in at least 20% of the malignant cells. Patients undergo leukapheresis for CAR T cell manufacturing prior to treatment. The study treatment begins with surgery for tumor biopsy or resection and catheter placement approximately 7 days prior to the initiation of cycle 1 of CAR T cell infusion. Patients are treated on one of 2 treatment arms, arm 1 with ICT delivery of the therapeutic cells, and arm 2 with dual delivery, where the cells are delivered ICT and intracerebral ventricularly, or ICV. Following a preliminary safety lead-in which patients are treated arm 1 and receive chlorotoxin CAR T cells through the ICT catheter only, patients will then be treated on arm 2, in which CAR T cells are administered both ICT and ICV via dual delivery, and dose escalation will proceed as shown in the table. In summary, chlorotoxin CAR T cell therapy is a novel immunotherapy that may extend the range of solid tumors targetable by CAR T cells. Results from our phase 1 study are expected to demonstrate the safety and feasibility of regional delivery of chlorotoxin CAR T cells in patients with recurrent glioblastoma. In this first-in-human phase 1 study, we will also determine the maximum tolerated dose and a recommended phase 2 dosing plan. This study is actively enrolling patients, and 4 patients have been dosed as of mid-April 2021. Thank you very much.